Okay, welcome everyone to day 3 of the DeGroote Center for Medicinal Cannabis Research, Innovations in the Science of Cannabis. Today is very exciting. We have our third keynote speaker, who I am delighted to introduce. Dr. Yasmin Hurd is the Ward Coleman Chair of Translational Neuroscience and the Director of the Addiction Institute at Mount Sinai. Her multidisciplinary research investigates the neurobiology underlying addiction and related psychiatric disorders. A major focus is directed on risk factors for addictive disorders, including genetics as well as developmental exposures, such as cannabis. But she's also developing novel therapies for opioid use disorder. And it's not overstating things to say that her work has really been pioneering in this area. So I'm absolutely delighted for her to be joining us. Thank you so much. And we look forward to your talk today. Thank you.
Thanks, James. I really appreciate it. Obviously, let me make sure that let's get my slides going. Okay. And I'm going to move all our nice pictures. So, clearly this conference is of such importance, and the fact that our society is going through so many changes in regard to cannabis and the discussions that are there, and make sure I'm going to focus today on the link between cannabis and opioids. And just like in the US, you guys in Canada have experienced this opioid crisis that in large part started with, many people trying to alleviate their chronic pain, that then switched many over to heroin. And then now we have the poison of fentanyl that has really exacerbated this epidemic, or keeping this epidemic here. And in the US, at least, the economic burden for opioid addiction is enormous, or substance use disorders in general is, $1 trillion a year. And our healthcare system, specifically for treating opioids, is extremely expensive because it costs nearly 3 times that of treating other medical disorders. Also, the overdose deaths are still, we're still in this epidemic that many people don't really realize of drug overdose that's driven mainly by opioids because of this pandemic. But this opioid overdose death and the epidemic that we have is still here. And, the problem is still trying to find treatments for opioid addiction, since many people don't use many of those that are available, which are mainly opioid agonists themselves. And so hence trying to find other alternatives such as cannabis. But it's not just the US that has the opioid problem. As you guys all know fully well, Canada also has an opioid epidemic and a lot of deaths. So the question is how do we get out of there? And as I said, especially for this conference, many people who are suffering or have suffered from an opioid addiction, and those trying to, manage their chronic pain, the whole question has been, which path do you take? Do you continue to use opioids that have all the problems that I just talked about, including death, or do you switch to cannabis? And that has been, the yin yang, this back and forth now, especially as in the US, and obviously Canada has also legalized, the use of cannabis for medicinal purposes, but also the recreational use of cannabis. And this back and forth has been very obviously confusing to many in the public, the lay public, but also for clinicians trying to treat their patients, and also obviously and the policymakers as well. And so, the thing that when I look at cannabis, and I think what this conference is about on many levels is, how do we weigh the risks and benefits of cannabis? And there are many aspects that we have to consider. We have to consider the risk for, addiction. We have to consider the risk for psychosis, so anxiety, other psychiatric disorders. We have to consider aspects of the developing brain. And as you can see, many times both of these outcome items are listed under both the risk and benefit category. So, how do we make sense of this? And this is one of the things I think where we need research to provide such guidance. And it starts with the fact for me that, we have to always remember that when we use the term cannabis is a very complex plant. It's not this homogeneous, medicine that we have one thing there that we are talking about. It's a plant with, over 140 cannabinoids that have varying degrees of psychopharmacological activities. It has other chemicals as well that may, yes, interact with the cannabinoids to either increase or actually even decrease their functional activity. But when we talk about cannabis, in large part, the public mainly, and I think rightly so for the recreational cannabis that's out there, they mainly think about THC because, again, all of THC is the main psychoactive cannabinoid in the cannabis plant. And that's the cannabinoid that leads to the euphoria and the high that why people consume cannabis recreationally. But it's really critical to understand not just THC, but where it binds or what does it do in the body. And this is where the endogenous cannabinoid system comes in. And many people always say, they smoke cannabis because of, it's mimicking our natural endocannabinoid system. And we have cannabinoid receptors where THC binds throughout our bodies, both in the brain and in the periphery. And in the brain in particular, as a neuroscientist, it's critical to understand that the endocannabinoid system is quite complex. In the brain, it regulates synaptic plasticity in large part through the cannabinoid receptor, primarily the CB1 receptor. And what it does is just modulates how other transmitters are released. But the endocannabinoids that are, or we could say, our natural, quote unquote, cannabis per se, it's like these lipids, anandamide and 2-AG, 2-arachidonyl glycerol. And they're synthesized by certain enzymes and metabolized by others as well. And it's this intricate, I would say, symphony of enzyme activity and stimulating these cannabinoid receptors that modulates normally our endocannabinoid system. And now THC specifically is a partial agonist at this cannabinoid receptor that can then modulate that activity. But it's important to understand that it's not just in the adult brain that the endocannabinoid system plays an important role. It plays a critical role during neurodevelopment. So the endocannabinoid system, they're endocannabinoids that, in terms of both, again, the enzymes that synthesize and degrade our natural endocannabinoids, and they play a role in the formation of neurons and glia, how the cells migrate, and indeed the synaptogenesis, the formation of these synapses. Throughout development, it plays a role even in synaptic pruning in terms of now fine-tuning the circuitry. And finally, it plays an important role in the maturation of the prefrontal cortex, that the prefrontal cortex is one of the last regions of the brain to achieve full adult maturity. So the endocannabinoid system is really important. So when we talk about cannabis and we try to liken it to our endocannabinoid system, that to me is where we need to make sure that we educate people, because the endocannabinoid system is about modulation, really fine-tuning modulation. But we're talking about cannabis, and in particularly THC, where it's not this physiological levels of tweaking our endocannabinoid system, it's high potency THC. And we know that, sadly, across the decades, the amount of THC in the cannabis being consumed today has increased from like 4% in the 1990s to going to 10% in here's 2008, and even today.
That's it.
Okay, So yeah, so I mean, I'm going to go quickly. This is just showing what I said in terms of overdose deaths and so on for the crisis and Canada, and the decisions that we're all making in regard to cannabis being, the risks and benefits that we are considering about cannabis. Just go the amount of cannabinoids. So this is where you guys, so just showing again, just going back that this is the endocannabinoid system. It's quite complex in the synapse in regulating and modulating how cells speak with each other by the natural endocannabinoid ligands, such as anandamide and 2-AG, and the enzymes that synthesize and degrade them. And this is just, again, showing the endocannabinoid system during neurodevelopment, that it plays a really critical role. So I think this is probably where you had stopped me. So again, like I said, I wanted to emphasize the aspect of when people talk about cannabis and being, a natural, our endocannabinoid system. It is not. And it's not because of the high potency with which it mediates its actions. Our endocannabinoid system, as I said, is a modulatory system. It fine tunes how cells speak with each other. And we have gone from like 4% of THC to now even 24% THC in the plants recreationally on the streets today. But I will say that even dispensaries and so on, there's no regulation about the amount of THC. They're products that are even 70% THC, and when kids and so on dab, there's even 80% THC. So there's no way that this is any way, shape, or form comparable to the modulatory system of our natural endocannabinoid system. So what about cannabis and opioid vulnerability? So one of the things, again, when we talk about decisions that people make, people are thinking about cannabis as a potential treatment or that it's better than using it versus heavier drugs like opioids or cocaine. And as I just mentioned, many people say, oh, cannabis is natural, it's just our natural endocannabinoid system. And that it, even now it's being promoted to pregnant women, that it alleviates nausea and morning sickness. But it comes with the corollary of, we know that substance use disorders like opiate addiction, that's similar to other psychiatric illnesses, they have a genesis in early development. While we may diagnose people in adulthood with substance use disorder, their vulnerability began much earlier in life. And that becomes a problem when we see this increased cannabis use in younger people, including pregnant women. And we know even that about 90% of adults with substance use disorder, they actually initiated cannabis use during adolescence. And on a biological level, the cannabinoid system, the endogenous cannabinoid system, overlaps significantly with the opioid system as well. So I'm going to talk and just show to you, some of our research that deals with the aspect of vulnerability for quote unquote cannabis in relation to the developing brain from prenatal and adolescent exposure, and what that might mean to the adult brain. And in regard to prenatal cannabis exposure, from early, and I'm showing you just the background, we mapped the cannabinoid receptors in the human fetal brain. And in the adult brain, the cannabinoid receptor, the CB1, the most prominent cannabinoid receptor in the brain, is actually the most abundant receptor that is on cell surfaces in the brain. It's the most abundant as compared to all the other receptors. During fetal development, it's expressed in areas that we call the limbic part of the mesocortical limbic system that regulates like emotions and so on later in life, like the nucleus accumbens and amygdala. And a lot of research has shown that the natural endocannabinoid system is critical for hardwiring of the brain in terms of the circuits and so on that lays down the tracks of our neurocircuitry. And we can see that these particular molecules, these binding proteins that are important for that cytoskeletal organization, those are significantly altered in the brains of fetuses whose mothers smoked cannabis. We could also see that transmitter systems that have been highly implicated in addiction vulnerability in adulthood, for example, this is the dopamine D2 receptor, and dopamine is a critical transmitter system for reward. And the dopamine D2 receptor, reduction of dopamine D2 is very characteristics of adults with substance use disorders. And there's always a debate about the chicken or the egg, which came first, the use of the drug or that the person had low D2 to start with. But here in the fetuses whose mothers smoke cannabis, they already have a reduced dopamine D2 that puts increased, would be associated with increased vulnerability. Also, they have reduction of this gene, the dopamine D2, in the amygdala, a region really critical for emotional regulation. Here you can even see sex differences where this alteration is driven more by the boys. So we know that the cannabis per se, at least is associated with these changes in the fetus. But in order to really get a better sense about the specificity of the changes, because many women could use other drugs and their life experiences and so on could make a difference. And the big question for us is really, what are the long-term consequences? It's not just being exposed to the drug, can it have long-term consequences on behavior? And so this is where we have our animal models. And I'm just covering a lot of different, research to say that we actually can replicate in our animal models what we see in human in terms of the fetal developmental stages. For example, dopamine D2 is indeed reduced by THC and not other drugs that we just to show that it was THC. And importantly, these impairments of dopamine D2, they last into adulthood, even though they never see cannabis THC again. And we can show changes in the functional activity of their cells and so on. So what happens to behavior? So if we let animals determine how much, for example, heroin they will self-administer, does their early life experience make a difference? And indeed, we could see that adult animals, when given access to heroin, they would self-administer more heroin. Importantly, even when we made the conditions so that they would take the same amount of heroin, those adult animals ran to the first hit, to get the first hit of heroin much faster than control animals. And whenever they're stressed, they self-administer more heroin. Importantly, they continue to show behavioral phenotypes predictive of addiction risk. For example, they have increased motivation to self-administer a reward. Here's a chocolate. They have to press either twice to get one chocolate pellet, then they have to press 10 times and so on and so on. And these animals will press over 100 times just to get one chocolate pellet. They also show greater depression-like phenotype and also anhedonic state, again, predominantly with stress. So the animal models were important for us. So we knew that THC itself could induce these long-term changes. But we want to go back and see what's really, can we study something in the human during early development to give us a greater insights, again, biologically. And it's challenging to look at humans longitudinally with prenatal cannabis exposure, but with my colleague Yoko Nomura, that's one of the things that we've been trying to do, and looking also in combination with stress. And we use a particular storm. Sadly, we had this hurricane in 2012 in New York. You can see New York is underwater. And we were able to, because we've been studying many women before, we were able to look at pre and post the storm as to their children and their offspring. And we've been studying many different things. I'm just going to move forward. So one of the things we wanted to look at is, does the early life, how can we get insights into what the fetus experienced in utero? And so we were looking at the placenta to give us some clues as to the impact of cannabis in early life during fetal development. And so we looked at the placenta and we've done molecular studies. And it just will show a few quick things. First of all, we could see that maternal cannabis use does change some of the endocannabinoid markers. For example, this is the gene that expresses the cannabinoid receptor. It's reduced in women who use cannabis. And for those women who we know they report their cannabis use, it's the more cannabis use, the greater the reduction of their cannabinoid receptor in the placenta. But what we saw that was significantly changed was really immune-related gene expression patterns in terms of the transcriptome. So immune dysregulation, whether in their cytokine-mediated signaling, interferon signaling, the placental changes were really striking in this immune dysregulation and in the women who smoked cannabis. We also saw that there was a double hit. I'm gonna go past this in terms of that those who had increased stress plus had smoked cannabis, whatever we looked at, whether it's their stress receptor gene or the cannabinoid gene, that the double hit had the greatest impact. But as I said, the question is about what about their phenotype. And so when we look at the kids in their offspring, of the phenotype of their offspring, we could see a number of things. One, again, this double hit that these kids showed much greater anxiety and aggression. But if I focus just on cannabis, we could see that there was increase in cortisol levels, the stress hormone levels in the hair of their kids. And as I mentioned, that there was greater anxiety, greater aggression, and hyperactivity in these kids associated with the maternal cannabis use. The important thing is we wanted to see whether or not the in utero environment predicted anything of behavior. And what was interested was, again, I'm only showing a snippet of our data, but we could see that the changes in the immune-related gene networks in the placenta actually predicted the anxiety behaviors that we see in their children, but it was only in those kids with maternal cannabis use. So absolutely, in terms of this question of, the yin and yang, maternal cannabis exposure, we do see leads to these protracted effects on their offspring and in terms of behavior and certain physiological measures, and many of those have been linked to the early onset of psychiatric and addiction risk. So what about adolescents? As I mentioned, we know, there's a lot with adolescent exposure that has been cited to increase substance use risk later in life. And in our animals, when we look at adult animals with adolescent THC exposure, we see that they do self-administer more heroin. And we can, again, go in and see what does it mean for the brain. So here, let's show some data regarding the prefrontal cortex, this region that I mentioned before, which is one of the last regions to develop to gain full maturity. And we could go in and we could look at the morphology of these cells, and we could see that these cells were actually the complexity of the cortical neurons differed. They have a reduction in complexity. And if there's a reduction in complexity, it means that the communication, the synaptic connectivity is weakened. Importantly, we could go in and we could use laser capture and pick up these cells and sequence them to see what molecular changes are occurring in these specific cells due to adolescent THC exposure. And surprisingly, there is a complete reorganization of the gene networks expression profile in these cortical cells as compared to the normal developing adolescent brain. They only share about 5% of the normal developing transcriptome. That's shocking. But where their change was interesting, they were changed once again, reflective of what we saw in the structural changes of the neurons, meaning, we could see that these genes related to the morphogenesis, but also there was a profound change in epigenetic modifications. And epigenetics is, how it's not changing the DNA sequence, but how the genes are turned on and off due to environment. And we can see again that while that both the genes that regulate synaptic plasticity and epigenetics in normal animals, there's not a great correlation, but there's a complete reorganization in the adults that had adolescent THC exposure. And it actually mimicked changes that we see in the prefrontal cortex of people who have been diagnosed, for example, with schizophrenia. So again, adolescent THC has these significant impacts on the brain. But as I mentioned, dose and potency of THC does matter. And we know, as I mentioned before, there's been this increase in THC across the years. So we've been looking at high dose THC. And here we, like postdoc Jackie Ferlin, looking at many different aspects of cognitive behavior, social behavior. We could see, for example, those adults with adolescent THC, especially high dose THC, they have greater impairments in social interaction, but they have a perpetuated elevation of their stress hormone, corticosterone, and even 95 days after their last THC. We normally see elevated cort in humans with THC, but this elevated with this high dose THC is really striking. And when they are stressed, here's a social isolation. The next day they self-administer, here's a chocolate reward, but they self-administer more. Their reward sensitivity is increased in the high dose animals. The high dose animals also show impairment of inhibitory control when they are re-exposed to THC in adulthood. I'm not going to go through the specific behaviors, but this is just a gambling task just to show that. So what is happening in the brain? Here we are looking at the amygdala. I'm sure just because I want to emphasize that there's a whole brain reorganization going on. And here in the amygdala, we could see something really fascinating. Mainly, a lot of times we studied neurons, but the cells that people used to call supporting cells, for example, glia, the astrocytes, we found significant changes in the genes related to these astrocytes. And the astrocytes are, in terms of the glia, these immune systems, the immune cells in the brain. So these immune cells in the brain are highly dysregulated with high dose THC, and especially with stress. And these cells are really critical for regulating synaptic plasticity. Interestingly, a marker of these cells, which is called GFAP, we see is elevated in these animals, and it directly correlates with their decision-making behavior. So the high-dose THC, in combination especially with stress, dysregulates the astrocytes, these immune-related the cells in the brain, and that correlates to impairments of decision-making. So it's not just prenatal THC, but adolescent THC exposure as well that alters the development of these mesocortical limbic regions. And it's not just neuronals, we see these, as I said, these immune dysregulation, and these are really critical for psychiatric and addiction risk. But dose, timing, and so on, we can go through in question period matters. I'm not going to go in terms of, we also see things lasting into the next generation. So I'll end with the yin and yang of the fact that most of the times when we study cannabis in our lab, we've mainly studied THC because that is the main psychoactive cannabinoid associated with addiction, for example. But there are other cannabinoids, and so that's why we had started to study CBD. And CBD, in the slide I had showed you about the potency going up over time, sadly, the potency of CBD had been going down in the plants at the same time. And research had started to show that, THC, as I mentioned, it's not just enhancing opioid addiction risk, but also even psychosis. And people had started to see that CBD was reducing psychosis risk. And so the question for us is, what would that mean in regard to opioid addiction? And when we gave animals CBD, we actually saw something very different. We actually saw that it reduced their heroin-seeking behavior. And what that means is when animals self-administer heroin, something goes on in their environment, whether a light or an odor, but it's a cue that's associated with them getting the drug. So after a while, you can just show them that cue and they will then start pressing the lever to try to get heroin. And that's what we call heroin-seeking behavior. And it actually decreased their heroin-seeking CBD. The thing that was interesting is that even 2 weeks after their last CBD administration, CBD was still reducing their heroin-seeking behavior. So there is a very distinct difference between THC and CBD on heroin vulnerability. And we can see on a molecular level that changes in the glutamatergic system, I'm not going to go into that, we see with heroin that we can replicate in animals that self-administer heroin, that CBD actually normalizes that. So that gave us hope that CBD might be a potential treatment for opioid addiction. And so we conducted double-blinded, randomized, placebo-controlled studies. And that's really critical. Without randomized placebo-controlled studies, it's very tough to interpret anything in relation to cannabis because, many people want it to work. And so in doing that, we look at people who were heroin abstinent, brought them in, and we showed them cues, just like we did to our animals, whether a heroin cue or a neutral cue. And those who had gotten the heroin cue, they craved. And CBD reduced that. And similar to the animals, when we brought them back a week later, after their last CBD, CBD was still reducing their craving. CBD works, as a number of other studies have shown now, on anxiety. And we showed indeed that when they got a heroin cue and had received placebo, they were more anxious, and CBD reduced that. And a week later, CBD still reduced their anxiety. But it's not just on the subjective effects. We want to make sure there are physiological effects that CBD is alleviating, and the stress and the cortisol hormone that is elevated with drug cues and stress, CBD reduced that. CBD also reduced the increase of their heart rate when they saw drug cues. So overall, we can say that the cannabinoid does matter. And here we can see that it's cannabidiol, CBD, that may hold promise for opioid use treatment, and particularly for our studies, at least for craving and anxiety. We still don't know the effective dose, the treatment regimen, and those are things that large clinical trials that we're on the verge of starting, we hope that will help to give us some insights. So if we come back to the yin and yang of cannabis, it's really about the cannabinoid. So yes, can cannabis itself have certain therapeutic risks and also benefits? Yeah, but when you're looking at a lot of the risks when it comes to at least addiction-related disorders like opioid use disorder, THC actually exacerbates that, as well as the anxiety, and definitely has a negative impact on the developing brain. But CBD seems to be beneficial for opioids, and other studies have shown now for some other drugs that are being studied as well. And obviously, you've also heard in terms of psychosis and anxiety, and obviously, you heard it earlier in regard from earlier lecturers in the conference about epilepsy. So the cannabinoid matters to whether or not cannabis is really therapeutic in a positive manner, or is it a risk? And these are some of the members of my team who have been really hardworking, and I mentioned some of them along the way. And Ed Salsis, who is an addiction medicine physician who is really outstanding, and the clinical team, and Yoko Nomura for our crazy longitudinal studies going from fetal development and studying the kids. And these are our great clinical team. And I'm glad to take any questions.
Thank you very much, Dr. Hurd. That's fantastic work. And I'm really, even after having looked at research in this field for a while, so intrigued by this difference that in the same plant you have two different chemicals that have two very, very different responses. Okay, moving to some questions. We have from Andra. Have similar studies on fetal brain development been done on other pharmaceuticals used by pregnant women, i.e. depressants, or used for the treatment of opioid use disorder, i.e. methadone? I'm curious as to how those changes due to cannabis compare to the alternative treatments.
No, those are important questions. I mean, clearly women, for example, who have an opioid use disorder and are pregnant women, we don't want to take them off their medication. So there are studies that have been, that have shown, and they've followed the children. And I'm not going to say that, that it's I'm not going to say that there's nothing that happens with those drugs in terms of their children, but their children are definitely better off with their parents being on the medication during pregnancy. And then, there are a lot of things that the children and help that they're given. but it's important to realize, many people, like I said, want to take THC and, cannabis instead, but the emphasis is that cannabis itself is not benign, even to the developing fetus.
Okay. A question from Margot. With CBD only exposure, are there the same anatomic and molecular level changes, longer term behavioral or psychiatric changes?
On a developmental level, we don't know. There are a few studies that are going on, if that's what you mean in terms of CBD, in terms of what are the long-term effects if CBD is given during the developing period. In our adult models is what we've studied CBD mainly because we're trying to develop it as a medication. And so that's why we've not seen any long-term side effects, but that's the whole reason of doing research so that we can try to now, the one good thing about humans is that we can actually ask them certain things as compared to our animal models. They mainly reflect the things that we study. So there may be other things that are happening that, we're not picking up in animal models, and the human studies will tell us. And there, as I said, there are a couple now preclinical studies being done on CBD and development. So we'll know soon.
All right. A question from Vic. What is the order of magnitude of CBD used in these studies? Tens, hundreds, thousands, milligrams?
Milligrams. So that's an important question. So to be honest, the clinical studies, I just went with what we had started studying in the rat, because at that time, no one was studying CBD. It wasn't like everywhere like it is now. So we looked at 400 and 800 milligrams of CBD. And the goal now is to start at the lower end and work our way down to see what may be the lowest dose that may be possible. But biology often works in like an inverted U shape for, optimal efficacy. So, we hope that we can gain some insights. And again, that's why you need research and clinicians don't know what to tell their patients, because the CBD that's offered on a nutraceutical level is nowhere near the same amount. They're usually 10, 20, 25 milligrams is sold in the stores, while clinically you're having, 100 milligrams, 400, 800 being used. So, so that's a big difference.
Yeah. Jason might want to comment on this. I remember a conversation with him when he was first planning some trials, and that the direction that he was told to go into was not to underdose for CBD in terms of the trials, that you don't want to run a trial and be underdosing the amount that you're using.
Right. And the thing that's tough is knowing what's underdose and what's overdose. Yep. And I don't mean overdose in terms of death, in the sense of, as I just said, often we see like this inverted use. So just because you use more CBD doesn't mean that you actually improve your outcome. In fact, you could be on the lower end going back down. And so you then lose seeing that efficacy. And that's why you need dose response studies. So, and it's tough to know where to start. So I feel for Jason. I know, felt the same thing. But I decided to just go with the sort of comparable dose that we had seen in the animal model.
James, did you want to comment?
If I could just offer a comment. I think that it's notable that a lot of the products that are out there are at much, much lower doses in the commercial marketplace, right? We're talking about tens of milligrams. And in our keynote speaker yesterday, I appreciated the point that he described those levels as almost homeopathic, right? You're not going to get a meaningful biological impact from 10 or 20 milligrams of CBD in that inverted U-shaped curve. You're just at the very bottom. So I think that that's a consideration in terms of thinking about the commercial marketplace versus medical interventions.
Exactly. And I want to add one thing to that. So, it's also important to realize we're talking about medicine for people with, significant pathology. Perhaps the commercial nutraceutical, somebody low level, whatever it may be, that 20 milligram may be fine, and it's not giving, just as long as it doesn't have, again, you got to make sure where you're getting your CBD, that it's really with GMP, manufactured in a good manner, and it's been shown to be safe, that it doesn't come with all, even THC might be put in there or other things. So, that's one of the things as well. I can't say that I know that for sure. I know that a lot of people tell me, oh, this has helped their anxiety, this and that. But I also know that those people don't suffer from the type of disorder for the patients that we're trying to treat. So could there be something? It may be, but without the research, like you said, James, you're not going to know if that little, is that little amount of CBD, if it's just at the tip coming up in that inverted U, is it enough for a certain type of problem? It might be, but it won't be for the people that we're talking about.
And just to weigh in on that, the way we've struggled with this issue is we've decided to allow patients to up titrate after randomization to the point where they are achieving a benefit, but without adverse events that they find problematic. So we're trying to account for the fact there could be some individual variability, there could be individual metabolic traits of patients. And so we're trying to give them enough, the option to go to a high enough dose that it should deliver a therapeutic effect, but to allow for some variability and then to learn from that. So we're initiating, if we can get permission to do our trial, which is another issue, we're trying to demonstrate feasibility and to then understand to what degree our patients up titrating and where do they end up lying in terms of that sort of final dose. But it is a real challenge, as Alan alluded to. We don't want to sort of provide too much CBD without cause to do so. But we don't want to run the risk of declaring a negative result simply because we didn't provide enough. So very interesting your work in this space. Thank you.
And I think that your model is a good one, in terms of, especially here when we really don't yet have so much knowledge about where to start. I think and randomizing as you're doing and having them titrate, I think that's a solid research strategy. So I think that, we will therefore gain a lot of insights. And we've done that with other medications, and people actually stop taking things, even some THC related products, when it was giving them, bad side effects. So then you start to see what concentration people are willing to take when the side effects start kicking in. And so definitely.
Thank you.
Great discussion. A question from Roman. Given the opioid craving reduction with CBD, would you anticipate a potentially opioid sparing effect in those with opioid treatment for chronic neuropathic pain?
Yes, and that's the goal as well. The one good thing about CBD is that it's the side effects that we've seen so far are really nothing, and it does not exacerbate any opioid negative effects. And the goal is to try to see if we can indeed reduce the opioid that's needed, even, aspects of methadone as well. Can we reduce the amount of methadone? Some people will always need perhaps an opioid substitution therapy, but the amount of that they may not need. And the same thing for pain management. So if you can reduce the amount of opioids, but still provide them with a medical benefit. So that's also one of the goals of CBD. So the fact that it doesn't interact negatively with opioid agonists is actually a very good thing, of being an adjunct, perhaps, medication.
This next question, I'm not sure if you're going to be able to talk about the Canadian context so much, Dr. Hurd, but perhaps in the US, if you have any knowledge about the US context. So the question from Elizabeth is Health Canada currently evaluating the information such as you present to consider capping the THC percent available recreationally? It seems like this could be a recipe for disaster where people can purchase high THC isolates with little to no protective CBD that could be epigenetically cause changes to the brain plasticity that cannot be reversed. Is health policy taking this stuff into consideration?
I from what I understand from my Canadian context, they are not. And the same thing in the US, which is a huge problem. We need to let the science and medicine inform policy, but instead policy actually ignored the fact that cannabis today is nothing like cannabis a decade ago, or even, like 2 decades ago and more. And this is the problem. It's not the same drug. If you have a 24 to 70% THC, it is not the same drug as when people like, the I always say the hippie generation and so on, it's very different. And also people back then were not consuming cannabis until a very till they were more like in adulthood. Now we have kids, early teenagers smoking cannabis, and it's with high THC. This is a problem. Not upping the amount of THC and the ratio of THC to CBD, I think, was a huge mistake, and not looking at the science.
Question from Kelly. It was stated briefly the increased risk for psychiatric disorders for children exposed during pregnancy. Was there any evaluation for autism?
There's definitely a huge battery of tests were done, and nothing with autism came up at all, or significantly at all, that could say what we showed was exactly what was the most associated: anxiety, aggression, and hyperactivity.
Question from Roman. Are there genetic chromosomal effects of THC on human gametes?
Only at ridiculously high concentrations that no human can really take. So, but it's other than that, no.
All right. From Nafisa, the argument sometimes used for keeping high potency cannabis is to compete with or stamp out the illegal market. But should we be competing with the illegal market when it involves selling such high potency THC?
And that's something for our society and policymakers to decide. And, at the end of the day, I don't think that we can ever stamp out the illegal market, no matter what you do. I think that, especially for a plant like, cannabis, people can always grow. There's always going to be that. The question is, what does a government promote? And if we're talking about health, you have to consider what you are allowing your population, your people, to be exposed to, that you're providing. And a government is supposed to really protect its citizens. And so you protect with knowledge. And that's the thing. We wear seatbelts because the knowledge was seatbelts help to protect lives. So it's the same thing in terms of mental health. I don't understand why mental health is not considered within medications being developed, but yet still we consider whether or not cancer may promote cancer, but we don't consider mental health. And I think that is a stigma that the government, every government, needs to get over. They're putting their citizens at risk, and especially their youngest citizens. I didn't mention one of the things about THC is that when it's exposed during adulthood, we don't really see the same degree of psychopathology as the early exposure. And so that's one of the things as well. But I just don't understand why mental health does not gain the same respect when we consider what is, medication development as in terms of quote unquote side effects.
Very good point. Question from Carlo. What do we know about the placental transfer of THC and CBD?
Unfortunately, cannabinoids pass across the placenta very easily. And that's one of the reasons why we said, okay, let's study the placenta, because it gives us some, could give us some information about the environment experienced. But it's also a problem because unfortunately they do cross the placenta, they cross into breast milk. So that's why we see more women also breastfeeding, not really good idea. So that's a problem.
Great. I think that might be all the questions. James, Jason, do you have any other final comments or questions?
I just want to say thank you again. It was an incredibly stimulating lecture, so wide-ranging, and it's been a pleasure having you join our conference this year. I hope you join us in the future.
Thank you. Thank you very much. Definitely, please keep having these conferences. We need education, and as data comes available, that's the main thing, good or bad. I think, I went down the path studying CBD, not thinking that it would provide this kind of insights, that it may be a treatment. So you have to be open-minded as a scientist, but you also have to look at the data, and hopefully the data helps to guide policymakers, clinicians, and the public. So thank you for all the things that you guys are doing. Thanks.